Good morning, students. Am I audible?
Yes, ma'am. Good morning, ma'am.
Good morning. So, we do the last topic in the diagnosis in theory part. Practical part, we are still going to discuss a lot of things. But in theory, the diagnosis of viruses will be done today. So, please let me know if I'm not audible because the network is really bad here. So, if you don't
Ma'am,
hear
you are
me,
audible,
Ma'am, you're audible.
but your voice is
okay, fine. If
quite.
I'm not audible, yeah, it's breaking, Devanshi. And that's why I'm saying if ज़्यादा खराब हो तो बताना because network I see Wi-Fi is very low here. Connectivity is low today. So the diagnosis part is will be done. So most, as I discussed yesterday also, that many techniques that we use for diagnosis are similar. So whether we are doing for a plant viruses, human viruses, or animal viruses, today we are going to discuss aquaculture or aquatic animals because these days aquaculture is also very important and these number of fishes and prawns and other things, these are being cultivated for being used as a food and in the other things. And they are also, viral diseases have been reported in all of them also. So, what kind of a diagnosis in those aquatic animals or aquatic we go for? So we are going. It is usually the same. We are following the RT-PCR or the real-time PCR approaches. We are following the serological approaches, and the basic approaches are same. But these days, the metagenomics or viral metagenomics is upcoming field where people, when they are working on a particular disease, they're looking for it as a most prospective approach for the novel virus identification. So yes, it is also being used. So diagnosis, yeah, animal diagnosis. Yesterday we discussed about molecular methods. We discussed about the serological methods. Microscopic examination or antigen detection is usually done. Then we discussed also about the latest, or we can say the newest approach is biosensors these days, which is also being used in diagnosis of the animal disease. A lot of research and lot of biosensors are being developed by the people for the pathogen identification. So this approach still under the study. People are still working under this. Otherwise, yes, microarrays, sequencing, RT-PCR, PCR, or a multiplex PCR, all these approaches are being already commonly used, right? So when we talk about the aquaculture industries, so there's a lot of expansion in this industry, and whenever when production is being increasing, or there's a we are focusing on the large scale production of anything. Along with that, the pathogen also there, right? So every now and then we keep finding new viruses or a new pathogens or viruses incidents or infection in maybe in a small amount, or maybe to the amount that the economic losses are also being reported is being, they came into in the form of a different research articles or a different report. So emerging viral pathogens, they are definitely a challenge to use a traditional uh, cell culture or immunoassays for identification of the new viruses. As I told you, the PCRs or serological approaches can only be used if the those pathogens which are already identified. In case of a new novel viruses or a new pathogens are there, so these approaches will not work because we cannot design the primers for the unknown viruses. We can only design primers for the known viruses, and we can identify them in PCR. Similarly, antibodies can be raised for the known viruses only. So all these approaches are good for the known viruses, but then for unknown and new viruses, these approaches somewhere they cannot be used, and that is one drawback. So viral metagenomics has potential to identify novel viruses where we don't need any knowledge on their genomic sequence data, and we can identify a vast number of the pathogens or viruses which are present. So metagenomics, high throughput next generation sequencing, and library construction. Lot of metagenomic projects are going on for the identification of the viruses. Also, these kind of studies are being done already. People are working on this, right? So when we talk about metagenomics, so what does metagenomics means? Have you heard this term before or not? What is metagenomics? Anyone?
Is it the study of the collection of
Yeah, it is. It is basically, yeah. It is yes, it is the collection of a genetic material or a genome from, number of community of organisms. So usually what happens is metagenomics studies, we don't rely only on the host, like the host of a particular pathogen. We study our outside that host also from the environment. Samples are collected from the soil, samples are collected from the water bodies, number of places the samples are being collected in the metagenomic study, and then those samples are being analyzed through number of the sequencing methods which are available. So next generation sequencing is the tool which is used in the metagenomic studies, right? So in terms of diagnostic studies, viral metagenomics has definitely a potential to serve as a multifaceted tool. They can study also, and they can identify the etiological agents of a single infection, or if there's any co-infection, a number of, whether they are aquatic organism or any other system, whether you want to study a phylogenetic analysis, we want to go for, or a genome diversity also, if you want to study the genome diversity also. So this kind of a study is very important. Traditionally, if we talk about the diagnosis of viral diseases, again, the cell culture is being used in case of aquatic animals also. And then we have a PCR also in aquatic animal, polymerase chain reaction, which is being used. Virus protein culture has been contributed to the development of many vaccines also against fish diseases such as pancreatic necrosis virus or viral hemorrhagic septicemia virus. So however, the bulk of viruses which are found in aquatic environment are unculturable. We are not able to culture them, and we don't, since they are not being identified, they are not being cultured. We don't have any antibodies which are available for their identification. Neither, since we don't have any genomics data, so we don't have any primates also, so that we can go for a PCR. So the detection of such non-culturable viruses by molecular biology-based tools such as PCR test demand that a genomic sequence of the target virus should be known prior to diagnosis. So which somewhere which is a hindrance basically, where novel viruses cannot be identified if their sequences are not known. So the aquaculture is continuously faced with the challenge of identifying novel pathogens or number of the infecting viruses which infect the fish or maybe the shellfish or crustaceans that we are using. So definitely there is a requirement where we need to develop diagnostic tool to identify these viruses, because diagnosis is a prior requirement for management of any kind of a disease. So correct diagnosis or reliable diagnosis are very important. So discovery of new viruses using viral metagenomics is being used. Then diagnostic role of a viral metagenomics in clinical tissues is also being studied, right? So in viral metagenomics, the goal is to ascertain the viral diversity in the environment. So that is often being missed in the studies targeting specific host potential reservoirs. So basically, when we look for the pathogens, we look for the host or a specific host. We try to isolate the pathogens from those particular host only. We focus on the other places than the host also, like in the environment also, in the soil also, the nearby areas also. These pathogen can be found. So this uses again the form to identify the new pathogens or viruses in the aquaculture, right? So this is just a small case study where influenza virus diagnosis. So how we go for the diagnosis of influenza virus. So normally, influenza viruses are the seasonal viruses. So not testing is not required for clinical diagnosis, right? So if they suspect some people with the influenza viral infection, they are given some fever tablets because we don't have vaccines, we don't have antivirals available for them, right? So they are given some proper treatment for a fever and other things. And we know that a seasonal flu is a very common problem, so usually diagnosis is not required. But virus testing can be inform the clinical management if the results may influence that. I mean, it is required for research purposes, or symptoms which are different than the earlier symptoms which were observed in case of influenza viruses are being noticed, then we can go for the diagnostics and we can go for the testing. So, testing is usually, if we feel that there is some outbreak of a particular disease in a particular area, then community centers and some camps are being set up by the government, and they go for some kind of a testing. So testing could be the antigen detection or rapid test, we call the rapid test also, or the more reliable one is the real-time PCR or a PCR that we use uh, for the confirmation of the virus in the population, or the number of the people if they are infected more. So diagnosis, see, sometimes diagnosis can vary with the viruses also. Like for one virus, we might prefer that antigen detection is more suitable, or other virus we might say that in this virus the result of a real-time PCR or PCR are more suitable. So we need to somewhere standardize the diagnostics also according to the viruses that we are working, different viruses that we are working, right? But the major diagnosis that we use is the serological and the molecular. Serological, ELISA is the major one that we are using. Molecular, the PCR or a real-time PCR is the major one. So other, when we talked about microarray, yes, they have been used in some cases, or hybridization, they are being used in some cases. But when we talk about the most common one, then serological, we talk about ELISA, and molecular, we talk about the PCR or a real-time PCR. Then multiplex PCR, yes, in some plant pathogens, the viruses, the multiplex PCR has been centered. But the commonly or the routinely used methods are ELISA and the PCR, which is being very commonly used for the identification or diagnosis of the pathogen, right? So this was all about the diagnosis. And with this, we finish the diagnosis part. And the next part of this module, or we are actually about to finish the course also very soon, because the course was not that much, but still I have selected some few topics, like we'll discuss about some sub-viral agents such as prions or viroids also in little detail, maybe one lecture each, or virophages or a baculoviruses, or what are multi-particles or a satellite viruses. So we'll try to discuss those, but after we are done with this. So the management part will take up next, and in the management part we'll discuss about them, because the management of a plant virus is totally different than the management of a human or an animal viruses. So that will take maybe 3 to 4 lecture on the management of a plant, human, animal viruses. So that I want to start in the next lecture. So today's lecture, we are done with the diagnosis. If there's still any questions or anything, you can ask your questions. Otherwise, we can just go for some short discussions on the topic. Yes, Yukta.
Um, Ma'am, you said traditionally the diagnosis of viral disease has been dependent on cell culture, but you also mentioned that in aquatic environment, a bunch of viruses are unculturable. So I didn't get.
Yeah, see, that is unculturable, means they are not able to culture number of the viruses which are present in the aquatic environment, right? So definitely the cell culture. See, I told you this thing also, that the diagnosis of the viruses can vary with the different kind of the viruses we are dealing with. It can vary with the aquatic, it can vary with the human or animal. But the major one used are ELISA and the PCR that we use. Culturable, yes, some viruses we can culture and some viruses we cannot culture. So that is the major problem that we face with the aquatic viruses, that we are not able to culture many of those viruses. So that is why they are saying the metagenomics is, or viral metagenomics is the answer to this kind of a problem that was being noticed in case of aquaculture viruses.
Ma'am, yes,
Yes.
ma'am. Ma'am, so is it not even possible to culture them even if we create optimal conditions in the lab?
Maybe, see, that's what this is. I've taken this from one basically paper. I'll upload that paper also. So they just said one statement that it was not very easy to culture those kind of a viruses or aquatic viruses, especially. They didn't talk about the human or animal viruses, but only about the aquatic viruses. So if it is not possible to culture them, then definitely we need to look for some other approaches and see what happens. Sometimes, when we work on a different kind of a viruses, the importance of the host which the viruses is affecting is more. We work more on the human viruses and a plant viruses, more on the human viruses on a priority basis, because we don't want humans to get affected. A lot of viral disease are causing death and number of other conditions. So we want to stop that. So a lot of work is being focused on that. Second priority, plant viruses, yes, because of food. Then it somewhere depends upon the priority also, because since yes, aquaculture viruses, yes, we were worried because definitely they are providing seafood is somewhere a very important part of a diet in many countries. So definitely we are concerned, but then we look for another method, because instead of going of thinking of what other methods could be used for the culturing, we look for the another methods which can be used or which can be easier than the culturing, just the cell culture method. So they looked for another approaches like the metagenomics approaches. Metagenomics is not a very new approach. Yes, it's use or people, it's exploitation for the research purposes or identification purposes might have increased right now, but otherwise it is very old approach, and people have been using this since maybe more than like 20 years back. Also metagenomics word was there and was being used, right?
Yes, ma'am.
Yes, others also, please come up your questions. So let's make this a discussion session and we can, talk more about this.
Ma'am, you had said that ELISA would come under like a serological method of detection, right? So
Hmm.
antigen detection, what would that come under?
It is also serological, beta. Whatever we are, when we talk about the antigen antibody, it is all serological. Antigen detection also is being, it's a rapid test which is being used some when we talk about the influenza or other viruses. So we should have some kind of antibody available with us for that virus. Then only that we are able to detect the antigen, right? So they all are serological testing. But serological testing, the most commonly one is enzyme linked immunosorbent assay, which we commonly call. They all come under the umbrella of serological testing only.
Okay, ma'am. Thank you.
So diagnosis of a pathogens, whether they are human or viral, or they are viral pathogens or a fungal pathogen or a bacterial pathogen, the diagnosis is very important, because without proper diagnosis we cannot think of the management of a viruses or any pathogen. So correct diagnosis will somewhere lead to the good proper therapeutics or a proper use of a therapeutics, or it will design the path of the treatment basically. So definitely a lot of emphasis is being laid on the diagnosis of a pathogens, diagnosis, correct diagnosis of the diseases. So diagnostic methods and the choice of a diagnostic method and which method we should choose for which kind of a pathogen is definitely somewhere very important, and it decide the path of treatment, and it decide the further, because if once we identify some pathogen wrongly and we are treating for some other wrong pathogen which is not even present in a human, even in a plant or in animal, so definitely we are not going to get any kind of a benefit out of it. So correct diagnosis is definitely very important when we talk about the management of any kind of a disease. So management, I thought that initially I thought start today, but then okay, let's leave it to the next lecture, because management is again altogether a new topic. And then when we talk about the management, as I told you, the management of a plant virus is very different than the management of a human viruses. For viruses in humans, management, the vaccination, we still rely on the vaccination. Yes, there are some of the antivirals which are available, and some kind of antiviral treatment for some pathogen, some viral pathogen is being recommended, but still We rely largely on the vaccines for the major or the deadly viruses. If we are being vaccinated, we can prevent the infection. And after management, the last step is usually eradication. So diagnosis, uh, treatment, management, and eradication. So once we know the management also, then we think of the practices at how we can eradicate this virus from the population. Now, for example, the polio viruses has been smallpox viruses. They are all eradicated viruses. Yes, still the cases, some cases in a smaller amount could be found there, but we cannot say that the outbreak is there. They are being eradicated, and maybe the major use of vaccination programs or immunization programs which were run by the government. So this is how it follows: diagnosis, treatment, management, eradication. So this is how. And when we talk about the plant virus management, it is very difficult to manage plant viruses once they are in the field. So a lot of steps in the viral disease managements are required to take before we grow those plants in the field. So before, what precautions or precautionary measures that we can take so that we are not introducing virus in the field. So mostly we are, once it is in the field, we cannot do anything. So what is recommended in case of a severe infections? You uproot the plants, you just destroy those crops which are totally infected. If you think that at the end the quality and quantity will be affected, or if it is in some for example, citrus tristeza virus was found infecting a lot of citrus trees in India also. So older people were recommended to uproot or destroy their older orchards and start with a new virus free kind of a planting material, or use some other methods for cross protection is also one technology which is being used in a plant virus. So I'm going to discuss the cross protection also in one lecture, that how cross protection is some kind of a immunization which is being provided to plant by using a mild strain of that particular virus, which will protect against the severe strain. So that technology has also been exploited in many cases. So there are a number of techniques that we use in a plant also, and number of techniques that we use in a human or animal plant medicine. So I guess let's stop this lecture today, and we can continue the viral management part in the next class. So yesterday also I was Devanshi, you were in the campus yesterday? Devanshi? Devanshi, can you hear me? I cannot hear you, but
Ma'am, am I audible?
yeah, now you're audible. You were campus में थे आप कल? You are again not audible, Devanshi.
Am I
No, you're not audible again. Yesterday I saw the assessment part. Athoiba did not turn on. Devanshi, Kshitij,
ma'am,
Nehal Joshi ने तो देखा भी नहीं.
ma'am, yesterday I was
You were not. Vivek also not turned in for yesterday. आप लोग कैंपस आते हो तो मिल लिया करो यार, इसमें क्या हो गया? बाहर से निकल जाते हो?
ma'am आए थे but entry नहीं मिल रही.
तो मुझे कॉल करते, मैं एंट्री लाता. एटलीस्ट यू हैव कॉल्ड अस. एंट्री ऐसे कैसे मिल जाएगी?
Ma'am,
आप लोगों ने ऑफलाइन ऑनलाइन है ना, आपने ऑनलाइन चूज किया तो कैसे मिलेगी एंट्री, बताओ?
Yes, ma'am.
आप सबको ना ऑफलाइन चूज करना चाहिए था. अब आप लोग सब आ जाओ कैंपस. आई फील इट्स हाई टाइम, राइट? So Nehal was also with you, right? Probably, and Vivek also.
No, ma'am, they were not with me.
Really? Okay, Devanshi, let's leave it here. Okay, students, so we'll stop here today. Let it be a small lecture, and maybe day after tomorrow we'll start with the diagnostic management part. Diagnostic we are done with, and the practical will take up another diagnostic part, right? So please, for attendance, switch on your cameras. Adhish Mishra.
Present, ma'am.
Yes. Akoiba. Yeah. Ayushi.
Present, ma'am.
Sam.
Present, ma'am.
Devanshi. Ishita,
Present.
Janvi, Manaswani, Namrata,
Present, ma'am.
Nandini,
Present, ma'am.
Nehal, Rupal, Sahiba, Sanjana,
Present, ma'am.
Shakti, Shivani.
Am I audible?
Yeah, you're audible. Tanvi,
Present, ma'am.
Tridiv,
Present, ma'am.
Vivek, Yukta,
Present, ma'am.
Yuvraj. Okay. So students is just okay. जितना भी हम we are almost will be covering up within this month or maybe the first week of October will be done with the syllabus. So after that we'll start the revision part also, and we will pick up some interesting topics that we can discuss further. So just try to go through the things that we have already done, and if required, so those things we can take up more in the revision section, right? So we can take those things, we can discuss those things more clearly again. We can take up some review articles, we can take up some case studies in case of a viruses, different case studies in a plant or animal viruses or human viruses. So I guess by October 10th, after that we'll get a lot of time for a revision and other things also, right? Okay, students, thank you so much. I'll see you day after tomorrow then, right?
Thank you.
Thank you, ma'am.